If you lose a finger, you don't grow it back. If you tear a ligament, it scars over. If you lose a limb, it's gone. This has been one of the most fundamental, assumed limitations of being human. Salamanders can regrow entire arms. Starfish can replace lost legs. But mammals, scientists for generations agreed, we simply cannot. That ability was considered evolutionarily lost, gone. A biological door permanently closed, but a study published this week in Nature Communications is challenging everything we thought we knew. Because scientists have just unlocked something hiding inside the body's own cells, something that suggests the door was never actually closed, it was just switched off. And they may have just found the switch. To understand why this matters, you need to know what happens when a mammal gets injured. The moment a wound occurs, the body launches an emergency response. Fibroblast cells race to the site. Their goal is simple: close the wound, fast, stop the bleeding, prevent infection. And they do it brilliantly. But there's a cost. In sealing the wound so quickly, those same cells lay down scar tissue. A kind of biological patch. It works, but it doesn't rebuild, it doesn't replace. It just covers. Compare this to a salamander. When a salamander loses a limb, those same types of fibroblast cells behave completely differently. Instead of rushing to scar the wound over, they organize into something called a blastema, a temporary cluster of unspecialized cells. And from that cluster, a new limb begins to grow. Scientists had long assumed this was because mammals simply lost the genetic instructions for building a blastema. Over millions of years of evolution, the new research from Texas A&M University suggests otherwise. Mammals didn't lose the instructions, they just stopped following them. The team was led by Dr. Ken Munioka at Texas A&M University's College of Veterinary Medicine and Biomedical Sciences, working alongside researchers from Tulane University, Arizona State University, Stanford University, and the Ludwig Boltzmann Institute for Traumatology in Vienna. Their approach was remarkably precise. They took amputated mouse, digit tips, the equivalent of a human fingertip, and they applied two proteins in a very specific, carefully timed sequence. First, a protein called FGF2, fibroblast growth factor 2, applied to the wound after it had closed. This first signal essentially tells the fibroblast cells, don't scar over. Instead, cluster together, organize, and remarkably the cells responded. They formed something that behaved like a blastema, a structure that scientists had previously considered completely impossible in mammals. Then came the second signal, a protein called BMP2, bone morphogenetic protein 2, applied to that newly formed cluster. BMP2 told the cells what to do with their potential, and the cells began to build. What grew back was extraordinary. Not scar tissue, not a rough approximation, actual bone, actual joint tissue, actual tendons, and articular cartilage, the kind of tissue that lines the inside of a working joint. Two independent regeneration processes occurred simultaneously at the same wound site. One rebuilt the bone, the other produced a joint-like structure with features of a natural finger joint. The results were imperfect. The regrown tissue was not an exact copy of what had been lost. Dr. Munioka was clear about this. This is really a two-step process, he said. You first shift the cells away from scarring, and then you provide the signals that tell them what to build. But here is the truly remarkable part. No external stem cells were transplanted. No foreign material, no lab-grown tissue implants. Everything that grew came from the body's own cells, already present at the wound site, ordinary fibroblasts, doing something extraordinary. This discovery challenges one of the most fundamental assumptions in regenerative medicine. For decades, the field has focused on importing external stem cells into injury sites, building tissue in labs, printing it with bioprinters, transplanting it from donors. All of these approaches share the same core assumption, that the body itself can't do this, that it needs outside help. What the Texas A&M team found suggests something profoundly different. The body already knows how to rebuild complex tissues. The instructions are already written into the cells, already present at the wound site. They simply need the right signal, delivered in the right order at the right time. This is not a question of giving the body something it doesn't have. It's a question of asking the body to do something it already knows how to do, but has been quietly refusing for millions of years. So, what could this actually mean for people? In the near term, the researchers themselves are cautious. The study was conducted in mice. Human biology is more complex. There are more cell types involved, more signaling pathways, more variables. Dr. Munioka's team acknowledges that the regrown tissue was structurally complete, but imperfect. And scaling this from a mouse fingertip to a human hand or arm is a very long road. But the near-term applications are genuinely exciting. Even without perfect regeneration, this approach could dramatically reduce scarring after amputations, could improve tissue repair for people with severe joint injuries, for veterans who have lost limbs, for accident survivors, for anyone facing the prospect of a lifetime with damaged irreplaceable tissue. The team believes this is the most immediate and achievable application, and both FGF2 and BMP2 are already known to science. Already studied, already understood, which means the path to clinical application may be shorter than most breakthroughs of this scale. The bigger picture here is about something even more profound than regrowing a fingertip. It's about a fundamental shift in how we think about the human body. For centuries we've assumed that what the body can't do is fixed, permanent, evolutionary fate. Salamanders regenerate, we scar. That's just how it is. But the more science looks closely at our own cells, the more it finds hidden capabilities, suppressed but not erased, switched off but not deleted. The thymus, thought to be irrelevant after puberty, quietly predicting longevity. Regeneration, thought to be evolutionarily lost, hiding in fibroblasts, waiting for the right signal. Every time we assume a door is closed, someone finds a switch. If you want to follow stories like this, make sure to subscribe. We cover the science that changes everything you thought you knew about your own body. I'll see you in the next one. We always thought the door was closed. It wasn't. It was just switched off. And scientists just found the switch. If you found this story as mind-blowing as I did, make sure to subscribe. We cover breakthroughs like this every single week. The science that changes everything you thought you knew about what the human body is capable of. I'll see you in the next one.